All glory is to God. Welcome back to Use to Understand Biological classes. Today, this class we will see the pentose phosphate pathway. Normally, the glucose which enter into our cell that undergo the glycolytic cycle. At the end of the glycolytic cycle, the pyruvate is formed. The pyruvate then enter into the citric acid cycle. Finally, it forms a ATP molecule. But in few cases, the glucose 6-phosphate is formed in glycolysis cycle that converted into the pentose sugars. That process is called pentose phosphate pathway. Why this pentose phosphate pathway is important? Because the formation of ATP and the ADP, especially the nucleosides, is formed through this pentose phosphate pathway, and also the free radical which forms whenever the oxygen molecule is exposed to our cell. That free radical is encountered by a NADP plus that is synthesized in pentose phosphate pathway. Let me see what are the processes take place in the pentose phosphate pathway. The pentose phosphate pathway means the oxidation of glucose 6-phosphate into pentose sugar. This process is otherwise called phosphogluconate pathway or Pentose monophosphate pathway because more Pentose monophosphates are synthesized in this pathway. Let me see. In this process, mainly it is oxidation of NADP plus and it forms the production of NADPH. The NADPH is an important compound which determine or which helps to encounter the free radical which is formed by the oxygen is exposed to our cell. The pentose phosphate pathway normally contains two spaces. One is oxidative space, another one is a non-oxidative space. In oxidative space, the carbon dioxide is released from the glucose molecule and it forms a ribose 5-phosphate. In non-oxidative space, the ribulose 5-phosphate is converted into glucose 6-phosphate. This process is a cyclic one. It is again and again the ribulose 5-phosphate is converted into glucose 6-phosphate and it forms a oxidative space of the pentose phosphate pathway. Let me see the oxidative space of pentose phosphate pathway. In oxidative space, the first step is glucose 6-phosphate is converted into 6-phosphogluconolactone with the help of a enzyme glucose 6-phosphate dehydrogenase. Okay, the glucose 6-phosphate dehydrogenase enzyme involved and it forms a glucose 6-phosphate into 6-phosphogluconate. It is a first step. In this first step, the NADP plus is reduced into NADPH and one hydrogen ion also here released. In second step, the 6-phosphogluconolactane, which is converted into 6-phosphogluconate with the help of the enzyme lactonase. Okay, with the help of the enzyme lactonase, the 6-phosphogluconolactane is converted into 6-phosphogluconate. Then the 6-phosphogluconate is converted into ribulose 5-phosphate with the help of an enzyme 6-phosphogluconate dehydrogenase. It is a second step to reduce a NADP plus. It is a second process where the NADP plus is reduced into NADPH. Here, one carbon dioxide molecule is removed. So here the decarboxylation process is also occurred here. Then the 6-phosphogluconate is converted into ribulose 5-phosphate because one carbon molecule is removed as a carbon dioxide. Next, this ribulose 5-phosphate is converted into a ribose 5-phosphate. What is the difference between ribulose and the ribose? It is just an isomerase. You see, in the third carbon atom, the OH group is different. So it is just like the isomerase. The aldose group becomes a keto one. Sorry, the keto group becomes a aldose one. So there is a ribulose difference between ribulose and the ribose. Ribulose is a keto group. Ribose is a aldo group. So the ribulose is converted into a ribose. That means the keto group converted into aldose. Here the enzyme pentose isomerase is involved. Now, in this oxidation step, the overall equation for this oxidation step is 1 glucose 6-phosphate is involved, and 2 molecules of NADP plus is involved, and 2 molecules of water molecule involved. Finally, it produces 1 molecules of ribulose 5-phosphate and 1 carbon dioxide, 2 molecules of reduced NADP plus. That means 2 NADPH plus 2 hydrogen ion. Here the net result is production of NADPH, the reductant for biosynthetic reaction, and also the ribose 5-phosphate, the precursor for nucleotide synthesis. Here the net result, we got 2 important compounds. One is NADPH. The NADPH is a good reductant for biosynthetic reaction. What is the biosynthetic reaction? Mainly, whenever the oxygen molecule is exposed to our cell, it produces a free radical. This free radical is somewhat encountered by this NADPH, and the ribose 5-phosphate is involved, or it is used as a precursor for so many nucleotide synthesis. In second step of pentose phosphate pathway is non-oxidative space. It is recycle pentose phosphate to glucose 6-phosphate, because the pentose phosphate is converted from the glucose 6-phosphate. Again, this glucose 6-phosphate is produced from the pentose phosphate through this non-oxidative space. It is the overall explanation of the non-oxidative space. In non-oxidative space, the first step is ribulose 5-phosphate is isomerized. It becomes a epimerase of cellulose 5-phosphate. Then again one another ribulose 5-phosphate is involved, and the 2 ribulose 5-phosphate and also cellulose 5-phosphate is forms. It combined to form 2 molecules, 1 7-carbon compound and also 1 3-carbon compound. Okay, 1 7-carbon compound is a pseudo-captulose 7-phosphate, and the 3-carbon compound is a glyceraldehyde 3-phosphate. Okay, the enzyme transketolase is involved. Transketolase means it transfers 1 keto group to the aldose the keto acceptor. Then in second step, pseudo-captulose 7-phosphate with pseudo-captulose 7-phosphate is reaction with glyceraldehyde 3-phosphate. It forms fructose 6-phosphate and erythrose 4-phosphate. Here, the transaldolase enzyme is involved. Transaldolase enzyme means the enzyme which helps to transfer of the aldose to the aldose acceptor. So the fructose 6-phosphate and the erythrose 4-phosphate is formed. Here, the fructose 6-phosphate again converted into glucose 6-phosphate, and erythrose 4-phosphate is joined with another one cellulose 5-phosphate. It forms a fructose 6-phosphate and glyceraldehyde 3-phosphate, and again another one glyceraldehyde 3-phosphate is formed through the same process. Then these 2 glyceraldehyde 3-phosphate forms a fructose 1,6-bisphosphate. Then after that, from the fructose 1,6-bisphosphate, fructose 6-phosphate is formed. Then the fructose 6-phosphate directly transferred to the glucose 6-phosphate. This oxidative steps is again and again involved in this reaction. It is the overall non-oxidative space. Here you see the 6 5-carbon compound, the 5-carbon 1, 2, 3, 4, 5, 6. The 6 5-carbon pentose sugar is involved and forms the 6 5-carbon compound forms a 5 6-carbon compound. 1, 2, 3, 4, 5. 5 6-carbon compound is formed through this non-oxidative steps. Let me see how this non-oxidative steps is involved. The first step, ribulose 5-phosphate is transferred into cellulose 5-phosphate with the help of a enzyme ribose 5-phosphate epimerase. What is different between the ribulose 5-phosphate and the cellulose 5-phosphate? In ribulose 5-phosphate, in the 3rd carbon atom, the OH group is present in the left side. Sorry, right side in ribulose 5-phosphate, but in cellulose 5-phosphate, it is present in the left side. That is the main difference. For that difference, the ribulose becomes a cellulose with the help of an enzyme ribose 5-phosphate epimerase. In second step, It is a trans aldolase. It involves a trans ketolase. It is overall process of trans ketolase. It's a keto group. It's transferred to the keto group acceptor or the aldose acceptor. You see, the pink color, the pink color bond is accepted by R2 group, and the green color bond is accepted by R1. So it is the aldose and the keto groups are interchanged between the two molecules with the help of the enzyme trans ketolase. This enzyme is highly dependent by the TPP enzyme. That means thiamine pyrophosphate. Okay, the thiamine pyrophosphate. This enzyme is involved to transfer the aldo group into a ketose and also keto group into a aldolase. Let us see how this aldose group and the keto group is transferred in this reaction. First, the xylose 5-phosphate and the ribulose 5-phosphate. The xylose 5-phosphate is a keto group, and the ribose 5-phosphate contains the aldo group. So the enzyme trans ketolase is involved with the help of a tryptophan, the thiamine pyrophosphate, and is convert a keto group from xylose 5-phosphate to ribose 5-phosphate, and the aldo groups of ribose 5-phosphate is transferred to the xylose 5-phosphate. Then finally it forms a glyceraldehyde 3-phosphate and the pseudo-captulose 7-phosphate. These two reactions are formed. Then the pseudo-captulose 7-phosphate and the glyceraldehyde 3-phosphate. Here the trans aldolase is informed. The aldo group is transferred from the keto group. The aldo group of glyceraldehyde 3-phosphate is transferred to the pseudo-captulose 7-phosphate. The keto group of pseudo-captulose 7-phosphate is transferred to the glyceraldehyde 5-phosphate. Finally, it forms the 4-carbon compound of erythrose 4-phosphate and 6-carbon compound of fructose 6-phosphate. I hope this colored compound, you see, this is a keto group. This keto group is transferred to the glyceraldehyde 3-phosphate. Then glyceraldehyde 3-phosphate became fructose 1,6, the fructose 6-phosphate. And the aldo group, which present in the glyceraldehyde 3-phosphate, it is transferred to the pseudo-captulose 7-phosphate. Then pseudo-captulose 7-phosphate becomes a erythrose 4-phosphate. Then xylose 5-phosphate and erythrose 4-phosphate. It forms a glyceraldehyde 3-phosphate and fructose 6-phosphate with the same mechanism. With the help of the enzyme trans ketolase and by the compound the tryptophan, the thiamine pyrophosphate. The trans ketolase is an important enzyme which occurs the cofactor, that is thiamine pyrophosphate. Yes, this thiamine pyrophosphate stabilizes a 2-carbon ion in this reaction. Okay, that is very, very important. Yeah, it is the non-oxidative space is finished. Let us see the summary of this pentose phosphate pathway. The oxidative pentose phosphate pathway that bring about the oxidation and the decarboxylation of first carbon atom of glucose 6-phosphate and reduce NADP plus to NADPH and produces pentose phosphate pathway. It is a first step in someone. Then NADPH provides reducing power for biosynthetic action. So many biosynthetic actions which occur inside the cell, mainly because of this NADPH and rapidly growing tissue such as the bone marrow cells. These cells obtain the more number of the pentose phosphate protein because in the cell, for the production of RNA and the DNA, especially the nucleotide, the pentose phosphate pathway processes occurred rapidly. The first phase of this pentose phosphate pathway is consists of 2 oxidation, that is converted glucose 6-phosphate to ribulose 5-phosphate and reduce 2 molecules of NADP to NADPH. In second phase, it is comprised a non-oxidative step that convert a pentose phosphate to glucose 6-phosphate. In this process, it is a cyclic one. It is again and again occurring process inside the cell. In second phosphate, the trans aldolase and the trans ketolase is important one. In trans aldolase and the trans ketolase process, the cofactor thiamine pyrophosphate is important because without this thiamine pyrophosphate, the process cannot occur. This process is also reversible action and it forms the non-oxidative space it produces a reversible process. In this reversible process, it produces 3 carbon, 4 carbon, 5 carbon, 6 carbon, and 7 carbon sugars are produced. This pentose phosphate finally gives to a 5 pentose phosphates. So 6 pentose phosphates, 6 pentose phosphate sugar finally produces 5 pentose phosphate. Okay, the common assimilating action photosynthesis, the same enzyme catalyzed by reverse this process. In pentose phosphate pathway, what are the processes involved? What are the enzymes are involved? These enzymes and these processes are reversibly taken place in photosynthesis. So that process is otherwise called reductive pentose phosphate pathway. The photosynthesis mechanism to produce a sugar is otherwise called reductive pentose phosphate pathway. The conversion of 5 pentose phosphate to 6 pentose phosphate. But the entry of the glucose 6-phosphate either into a glycolysis or into a pentose phosphate pathway is largely determined by the relative concentration of NADP and NADP gas in the cell. So it also a regulatory compound. The NADP plus is called a regulatory compound because this NADP and the NADPH, the flux of this NADP and NADPH determine whether the glucose 6-phosphate is entered into a glycolysis or pentose phosphate pathway. I hope you understand very well about the pentose phosphate pathway. Do you have any doubt? You feel free to set in the chat box. I will clarify your doubt. Or if you want to explain in pentose phosphate pathway in details, please let me know. I will explain you in detail. Thank you.